Welcome everybody. Today we have the Bioexcel webinar 84, and it's on perspective on simulating whole cells with Martini. Our speaker is Siewert-Jan Marrink from the University of Groningen. I'm Alessandra Villa, and together with Otto Andersson, I'm hosting this webinar for Bioexcel, Center of Excellence for Computational Biomolecular Research. This webinar will be recorded, so you are aware of that. And during the webinar, you can ask questions using the function that you find at the bottom of the Zoom application. You can find this symbol or this symbol according to your operating system. And then you can type your question. You can type it if you want in an anonymous way, so we know that you don't have the microphone or you don't want to speak, or in also not anonymous. And then if you don't have the microphone, in that case, please tell us so we don't try to unmute you. If you have the microphone and you want to speak, so then we will unmute you to ask yourself the question. So after the webinar, you are most welcome, if you still have questions for Siewert-Jan, to ask the question using the Bioexcel Ask Bioexcel forum. Here there is the link, or you can just use the QR code, and then you go directly to the post of the webinar of Siewert-Jan, and then you can ask the question in the following week, and Siewert-Jan will answer to your question. So now something from the presenter of today. So Siewert-Jan was educated as a physical chemistry. He got his PhD in 1994. And in particular, in his specialization was the use of computational modeling. The central aim of his research is to unravel the lateral organization principle of biomolecular membrane and to develop efficient computational models to simulate molecular process. His group is well known not only for the Martini models, a coarse-grained force field for biomolecular simulation, but also because they have contributed in understanding membrane, in particular related to the process of fusion, domain formation, and membrane protein activity. Like is one can say, simulating is believing. Like Watson will say that see is believing. But Marrink is also credited for several methodological advances to improve the computational microscope, including development of pioneer multiscale simulation algorithm and high-throughput tools. And now I'm curious to hear what he will tell us about the whole cell. I stop sharing and I welcome Siewert-Jan.
Okay, thank you very much, Alessandra, for this kind introduction. Let me share my own screen, and hopefully you can see now already my first slide. Then let me see if
yep.
Laser pointer. Perfect. Okay. So yeah, this talk will be about the use of computational microscopy to simulate whole cells, because that is what we are currently aiming to do. And with the overall idea of that what I cannot simulate, I cannot understand. So simulation is, in my view, a very important tool to create understanding that is difficult to achieve with traditional experimental methods. And I think this highlight of a cell cytoplasm that you can see here already provides this idea of the power of the computational microscope, because you can see here the molecules at a level of resolution that is unmatched by any experimental microscopy techniques. And I'm not only talking about spatial resolution, but as you can see, we also have a dynamic resolution covering timescales from picoseconds up to milliseconds. So yeah, the computational microscope is actually a microscope that can or needs to operate at different kind of resolutions depending on the process that you're actually interested in. On the one end, we have the quantum mechanics, if you really want to study the chemical reactions that are taking place in a cell. And on the other end, you have continuum models that can really cover the length and time scales for cell-cell interactions, for example. But in between, this is the realm of molecular dynamics as one of the major simulation techniques. And in particular, our group is involved in this intermediate level of resolution called coarse-grained molecular dynamics, where you simplify to some extent the description of your molecules to allow for larger spatial temporal scales to be accessed. All right. This slide I include just to highlight the immense progress that we have witnessed in my own field, which is mostly centered around simulating lipid membranes. And as you can see here, when I was doing my own PhD, we were struggling to get simple model lipid bilayer stable that consists of about 60 to 64 lipids. And at that time, we were really trying to build force fields that would work for lipids in the first place. But then, as you can see, due to both hardware and software development that make the hardware and software much more efficient and faster, but also due to this business of coarse graining, we are now able to simulate these membranes at a level of realism that really matches those of real cell membranes, including a large number of different lipid components, as well as many of the membrane proteins that you will find in a typical, say, plasma membrane, and also interaction with the cytoskeleton underneath. So this is where we currently basically are, and then to the right, you see where we are heading towards, because the eventual end game of understanding a lipid membrane is to consider it in its complete native environment. As you can understand, this will be in the end important because everything connects to everything else inside a cell. So that's why we're now trying to simulate complete cells. And I will explain you in this webinar how far we are in this challenging endeavor. All right, but first a little bit about the essence of coarse graining. So the coarse grain model that we developed in our lab is coined the Martini force field. And it basically is a force field that instead of considering every atom in the system explicitly, it groups several atoms together with, so heavy atoms together with the associated hydrogens into coarse-grained beads. And Martini maps on average 3 or 4 heavy atoms into such a bead. And so by reducing the amount of particles that you have in the system, and also because you use a more smoothened potential energy surface, you can use larger time steps. And together this makes this a very efficient model for coarse-grained molecular dynamics simulations. So the current Martini dome, which is growing over the years, consists of basically all the biomolecular components that you need to simulate a cell. So we have coarse-grained models of water, of course, of all the lipids, proteins, sugars, nucleotides, but also the same force field can be applied also in the area of material science to all sorts of polymers and nanoparticles. And so the key features of this Martini model are that it retains chemical specificity. So despite that we coarse grain and we throw away some of the details of our models, we can still meaningfully discriminate between all the different amino acid side chains of a protein, or between whether a lipid tail is saturated or unsaturated, different head groups, different sugar moieties, the nucleotide bases, etc. So it has still this chemical resolution that we find useful. It's also fast, about 3 orders of magnitude faster than a traditional all-atom model. So that would be the main reason for resorting to a coarse-grained model to get the speed up. And then the way this model has been parameterized, and I don't have much time to go into that into detail, but the way we do it using a building block approach means that you can combine all these coarse-grained beads together, and the model is very compatible. So you can mix proteins and lipids and nucleotides, and all the coarse-grained bead interactions have been carefully balanced during the parameterization of this Martini force field. That makes it also a very versatile and pragmatic model. So if you're interested in including other type of molecules in your simulations, you can easily follow the kind of same parameterization strategies that we have outlined over the years, and you can mix them with all the other ingredients. So the parameterization, which is still ongoing, I must admit, because if you're in the business of force field development, this is basically a never-ending story. There's always ways to further improve the models, either because there's more powerful computers that you can use to test and validate your models, or because there's more experimental data available. But the basic interactions in Martini are a combination of a top-down parameterization, where we use extensively thermodynamic data and think, for instance, about partitioning of small molecules between different types of organic solvents. We use that to calibrate mostly the non-bonded interactions underlying the Martini force field. And Martini uses standard Lennard-Jones type of interactions to mediate these non-bonded interactions. And then we also rely on bottom-up approach, because we often use reference atomistic simulations on small systems to calibrate mostly the bonded potentials that we also need, the harmonic bond potentials, angle potentials, etc., that we use to keep our molecules in the correct shape and to give it a flexibility that would correspond to those seen at the atomistic level. All right, that's the Martini model in a nutshell. Of course, there's much more to talk about, and one aspect is that it's under constant development, as I already alluded to. So originally, in the early 2000s, the model was only able to describe lipid systems, but then it was extended and became more mature in the following decade. And then the Martini 2 model was released, which could treat basically any type of biomolecule. But then development went on, and in particular, Paulo Sousa, postdoc at the time in my lab, did a tremendous job in further calibrating the interactions underlying Martini 3 and making it even more generic. And so now we have the Martini 3 version of the model, which is really a general purpose type of model. So a couple of things that we have been improving since the Martini 2 release. So there has been a reparameterization of basically all the beat-beat interactions, and also the bonded interactions now using a center of geometry rather than a center of mass type of mapping. And this overall leads to improved interaction balances between all the different beat types, and also better packing of molecules together. Then we have introduced more beat types, and I will show on the next slide the family of beat types that we are currently having. More beat types and ways also to modify the interactions to allow for even a better coverage of the huge chemical diversity that we eventually would like to include in our Martini model. Then we also have reconsidered the way that we treat charged beats. So we have included more ionic types and also double charged ions, and looked at, for instance, Hofmeister series, see if we could reproduce these kind of known physical chemical effects. And then another important change with respect to previous models of Martini is that we now have Go models as the standard elastic network to keep the overall secondary and tertiary structure of proteins and also of nucleotides. Because one aspect or one limitation of Martini and coarse graining in general is that it has difficulties in doing, for instance, or simulating protein folding, as we in having these coarse-grained beads lack the anisotropic hydrogen bonding that is required to stabilize, say, secondary protein structures like an alpha helix or a beta strand. So we need some additional network to stabilize these configurations, and now we use kind of Go-type models to do that, which also still allows some flexibility and unfolding of certain regions of the protein. All right. although we are with Martini 3 at the moment, and this is our current workhorse, still Martini 2 is used by the community a lot, and we also use it. And actually, some of the simulations I will show later on the whole cell are still based on Martini 2. So it's still a very useful model by itself. All right, just beat it. So this is actually an overview of the current bead types in Martini 3. So we have multiple major classes of bead types, ranging from non-polar to polar charged, and a separate class now for halo compounds, and a number of subtypes that further distinguish these beads, allowing us to represent a large variety of chemical building blocks. They come in different sizes, so the standard 4:1 mapping requires a regular bead size, but sometimes you need higher resolution mapping. For instance, in planar parts of molecules, many drug-like molecules, they are planar, and then this 4:1 mapping is not very suitable, so we need somewhat higher resolution. And all these bead types have been calibrated with respect to each other, so you can mix and match them at will almost, although the central choice is still this 4:1 mapping. We have a specific bead type for water, which also allows us now to more easily implement different types of water models. And we have also now much more extensive bead label types compared to previous versions of Martini, not only distinguishing donor-rich or donor-acceptor or donor, hydrogen bond donor or hydrogen bond acceptor moieties, but also moieties, for instance, that are electron-rich or electron-poor, depending on the chemical substituents. Okay, that is, I think, what I'm going to tell you today about Martini. You can, of course, look up a lot more details in this Nature Methods paper. The main paper is actually an easy read. It's about 2 or 3 pages with some nice examples. But then actually the supporting information is about, I don't know, about 100 pages, I think. I don't think everybody or anybody ever read it completely, but if you make it to the end, there's actually a nice reward because there's actually a recipe of a real Martini cocktail to be found there. I'm not sure if the Nature editors realized this, but they published a nice cocktail recipe. Yeah, so summarizing, we now have about more than 800 different coarse-grained beads in Martini, with a total of 355,000+ pair interactions, because all the pair interactions are specifically parameterized in Martini, although I have to say immediately that a lot of that has also been done through extrapolation or interpolation, I have to say. So a total of more than 1,300 parameters underlying this Martini 3 model. All right, before I go to this whole cell, I read in my own abstract that I also promised to say a little bit about reactive Martini, because that's something we're currently trying to explore, to what extent we can actually incorporate some reactivity into our coarse-grained models without having to use, of course, a whole quantum type approach, because that would slow down the coarse graining so much that you wouldn't benefit from the coarse graining anymore. So we now have a pragmatic approach based on virtual sites that can be used to lock certain groups or reactive groups into a fixed configuration. So in this case, we try to model the formation of a disulfide bridge. So we have 2 sulfide reactive groups in Martini that each carry a number of these reactive or virtual sites that make sure that whenever these compounds get into close contact, they have a high likelihood of actually forming a stable bond at a certain angle, mimicking actually the reaction that would have taken place in reality. And the way we do this, so we have a kind of tabulated potentials between these virtual sites that allow us to have kind of an angle-dependent potential. So here you would see a standard Lennard-Jones potential that has kind of a minimum at a certain distance, and then a steep repulsive part here and a longer attractive well at the other side. But no matter what angle these 2 beads would approach each other, they would have the same interaction. That's the standard Lennard-Jones interaction. But now with these reactive potentials, you can actually make it such that at a specific angle, here it's at 0, but you can put that at any angle that you like, you can create a configuration. They can stably stay there for as long as you want, because you can tune, of course, the reversibility of this reaction by tuning this well depth. And you can also include a barrier such that you can also tune the reaction rate. By including a barrier, these particles will have a harder time eventually locking together, but once they are then locked together, they will also be kinetically trapped there. All right, so that's the idea. And we have tested this on a particular reaction where, as you can see here, we actually have a compound where there's 2 of these disulfide bridge forming units that can together allow the formation of rings. And this is experimentally done in the lab of our collaborator, Siewert Otto, here in Groningen. And we can now simulate that. So if we start with a box of these individual monomeric compounds, but now make these 2 beads reactive, then you can see the formation eventually of these rings of different type of sizes. And you can follow the oligomerization, and eventually we see that in this case, trimers of this ring as well as tetramers are the dominant species at the end of the reaction. And this is actually in line with what the experimentalists are also finding for this particular system. And here it's shown that you can, by introducing this barrier, you can actually tune the rate of the reaction, the rate at which the monomers are being consumed. So this is a very pragmatic way of capturing reactions. So I need to stress that the actual mechanism of the reaction, of course, is out of reach because we don't include any quantum mechanical details, but we can capture the rate and the reaction products. And what is important, and that is, I think, the main strength of this method, is that you can also now capture the effects of the environment. So you can embed these particles in different environments and see how that affects the rate and the products of your reaction. And you can also do this in a non-equilibrium kind of setup. It's very pragmatic. It doesn't require you to stop the simulation. This is just the topologies that you provide, and then the molecules undergo normal standard MD. So here's an example of where we have applied this now for the first time to again look at this ring formation, but now in combination with a peptide that forms a coarse-grained or a biomolecular condensate. So this is synthetic peptide synthesized by the Spruit Lab here in the Netherlands. And in accord with the experimental data, this peptide forms a nice condensate. And you can see here that we can already show by a simple potential of mean force calculation that these building blocks, these reactants, actually favorably partition into this condensate. So they naturally like to embed themselves in the condensate. And now we can see how the condensate actually has an effect on the formation of these multimers, these oligomers, these ring-like oligomers that result from this reaction taking place. So this is now a simulation, the end snapshot of a simulation where we put a lot of these monomers together with the condensate forming molecules. And then at the end, you see that both the reactants and the product end up inside this condensate, again forming these kind of ring-like compounds. And then here we actually compare the final oligomer size distribution between the system in the condensate and a reference system that I showed you before, where you have a purely aqueous environment. And so whereas in the aqueous environment, you have mostly the tri and tetramers that are forming, you see that in the condensate, you actually have a shift towards much larger ring sizes that are being formed. And again, this has been confirmed by our experimental collaborators in the Otto group. So I think this showcases the potential of this reactive Martini scheme. All right, so in this case, you can see that biomolecular condensates can actually accelerate the reaction rates and also tune the reaction products, mainly by concentrating all these reactants inside a smaller system or a smaller size. All right, then now to the main meat of this webinar, our attempts to model an entire cell. And if you want to model an entire cell, then you have to decide what cell you want to model. And being someone that is aware of how costly it is to simulate large-scale systems, we of course went for the smallest cell possible. And that is the engineered cell by the Greg Venter lab, which is the JCVI Syn3A minimal cell. So this is a cell that is derived from a mycoplasma, a bacteria stripped down from actually basic or stripped down from all the genes that turned out to be non-essential. So they have just deleted as many genes as possible until they had a point where the cell was not viable anymore, and they reintroduced these genes. So it's kind of a minimal life form. It's ideal for simulation not only because it's relatively small, as you can see, it has a diameter of only 400 nanometers, but also because the composition is almost fully resolved. And of course, we need that information to set up a simulation. Also, the envelope is rather simple. It's just a lipid membrane-based envelope. This particular minimal cell doesn't have a cell wall. So what does it contain? It contains 453 kilobase per chromosome, so a single circular chromosome coding for almost 500 genes. And then it contains about 77,000 individual proteins of 450 different types at a very crowded density. So this is similar to the most crowded bacterial cell concentrations. And the proteins making up more than half of the total biomass of this cell. Then it also contains hundreds of ribosomes and the RNAs, totaling about 16% of the biomass of this cell. It contains also millions of small metabolites for a total of 6% of the biomass. And then the envelope is composed of millions of lipids of a few different types that largely depend on the surrounding, the way this cell is being fed. So yeah, as I say, relatively simple, but still overall still a very complex system to be simulated. So if you want to simulate a cell like that, you have to use what we call an integrative modeling framework. You have to integrate data from many different resources. And so we have done that in collaboration with the lab of Zen Luti Schulten at the University of Illinois, combining actually experimental data, for instance, cryo-ET data that gives you an idea of the overall shape of the cell together with the distribution of the largest components. Cryo-EM data that gives you information about the various complexes that you might find in this cell. And of course, a lot of omics data that tell you which components are there and at which quantities. A lot of that data was already integrated in the work of the Schulten group because they are studying this cell at a much coarser level using kind of lattice microbe or mesoscale type simulations. And they are trying to follow in the reactions that are taking place inside these cells and predicting along the different stages of the life cycle of this cell. But they also needed all this information for their reactive models. So a lot of this information was already collected. So based on this, we then integrated this into our Martini ecosystem, a kind of collection of tools that are necessary to eventually build this entire model. And so a number of these tools are, for instance, Polyply, which is a tool that can very efficiently pack and generate or pack molecules together and generate starting structures of large polymers required for the chromosome. We have this Martinized tool that can build topologies for the proteins that we need. And we have a tool, TS2CG, that can build the membrane envelope and the included membrane proteins. And all this together, we have another tool called BentoPy, after the Japanese bento boxes that pack your lunch very efficiently together. This packs all these things together and can now create entire cell models in a very easy way, I would say. I think you can give a complete new list of ingredients, feed it into bento pie, and you have the starting state of your cell model within half an hour. So that really is not a bottleneck anymore. Okay, so here I take you through the steps of actually then building this initial model. So starting from the geometry of this minimal cell, we first place the ribosomes, which have been resolved also by cryo-electron tomography. So we know that they're more or less randomly dispersed throughout the cell. So we reserve the locations of the ribosomes. And then we first start to embed our chromosome inside the spherical volume, but avoiding overlap with the ribosomes. So we first did a supra coarse-grained self-avoiding walk to create the initial structure of our chromosome. Then we did some polynomial fitting, converting it to a 3D spline. And that would allow us then to back map the Martini level resolution nucleotide model that we have to eventually create this entire circular chromosome at the Martini level of resolution. Now, when I first saw this particular image, I was getting worried because I thought, okay, now we have the chromosome and the ribosome, but the whole thing is already completely filled. And we still need to add all the proteins and all the other stuff. So aren't we running into trouble? However, if you then zoom into this model, you can already appreciate that there is still a lot of empty space between the chromosome and between the ribosomes. So there's still space to put all the proteins eventually. And if you further zoom in, you can really nicely see the DNA strands here at the Martini level of resolution. Yeah, and we're really, every base, we really have the real base sequence of the real JCVI SYN3A model. All right, so having the chromosome and the ribosomes, we now have to put the envelope around it, constructed using this TS2CG tool. And then we have another tool, SoupMaker, that uses this Martinized tool in a very high throughput way to test and validate all the individual proteins that we need and make sure that the topologies are fine, either using known PDB structures or using AlphaFold, and also inputting known macromolecular structures composed of multiple proteins. So we can take all this information into account, put it in our pipeline, and come up with a nice crowded cytoplasmic solution. And then putting this all together, we created our first model of this entire cell in almost its full complexity. And this was published more than a year ago now in this Frontiers in Chemistry model. So, because I think normally you wouldn't just write a paper about building a system, but I think here the building was actually already a major effort that deserved its own publication. But of course, we want to simulate it. So we put it in a box of water, and you can see here that the total system size is more than 600 million coarse-grained particles. If you would want to do this at the atomistic level, you would have another factor of 10 larger system size. So this is already very huge. Yeah, as I said, it contains all the major components. However, it turned out that we were not able to simulate it. Of course, we tried loading this into GROMACS, our workhorse software, but GROMACS complained and said, look, this system is too big. I can't handle that. So then we contacted the GROMACS developers, in particular, Eric Lindau has helped us a lot with trying to get this kind of huge systems to run with GROMACS. So initially, the particle limit issue had to be resolved. So that has been done. Then we encountered some other issues because one of the major hurdles turned out to be that we have this very long chromosome, which is a single molecule, a single topology spread over multiple domains, and that gave all sorts of issues with GROMACS. Long story short, we're still working on getting this model to run efficiently. I think we're almost there. So soon we hope to be able to simulate this entire cell at its full size. But in the meantime, we actually started testing smaller scale versions and also looking at the individual ingredients in a little bit more detail. So here we have 1/10 of the total volume of the cell, looking at how 1/10 of a chromosome behaves dynamically. So here we just have the chromosome confined in a spherical volume. And then you can do some dynamical analysis looking at the correlation between the factors along the DNA axis. And if you have from the correlation analysis, you can determine the persistence length. You would expect some kind of exponential decay, but because we are now in the spherical confined volume, you get some kind of oscillations on top of that. But you can fit this, and you can fit this to a persistent length of about 49 nanometers, which is quite reasonable for a double-stranded DNA of this type. So that's one type of validation of our chromosome model. We also looked at simulations of this dense cytoplasm. So here you see, again, in a spherical constraint, 1/10 of the total volume of the cell, but with the full complexity. You see all the individual proteins, you see very tiny molecules, many of the metabolites. And if you zoom in a bit, you can appreciate more the already very intriguing interplay between how these proteins interact with each other, how the proteins interact with the metabolites, how the proteins interact with the chromosome, and how the metabolites interact with the chromosome. There's a lot of things going on, and we're still in the process of actually starting to analyze all this information. Because as you can appreciate, running these simulations is one thing, but the analysis of these kind of complex systems is by itself also quite a challenging and demanding task. All right, but that looks good. Then we're also considering the envelope of this particular minimal cell, which has a very peculiar composition. It actually is very much enriched in cholesterol, up to almost 60%, which is quite unique for a bacterial cell membrane. And the idea is that maybe this cholesterol is needed because this cell doesn't have a cell wall, so it gives a certain robustness to the cell membrane. It actually takes up the cholesterol from the environment, so it needs this in the medium that is surrounding this cell. And here you can see a simulation just of a bilayer patch of this particular minimal kind of lipid composition that this cell needs to survive, showing that indeed this cholesterol seems to lead to a quite well-organized and stable lipid membrane. And we can also scale this up to the level of an entire large liposome. But also, of course, we can look at all the individual membrane proteins that are part of this cell envelope to make sure that the individual proteins behave nicely, and also to get information on potential lipid enrichments around these proteins that can be used to better create starting structures of the entire model. So we can do this for all the different type of membrane proteins, get this information, and then use that to back integrate this information into building the entire cell. All right, so now we are ready to simulate at least 1/10 of the cell, because that turns out runs already well in GROMACS, so we have no troubles. And now you can sit back and enjoy the first simulation of an entire cell. And I can already assure you that this will be something that you've never seen before. This is already giving us unheard dynamics of a cell that we are currently trying to get some experimental validation. But I'll show you what you will see. Isn't that fantastic? Complete unexpected behavior. I must say I was quite fascinated by this. And I think we must admit that in this case, it might not be entirely realistic. unless someone can tell me that they have seen this in the test tube. This is what happens if you start simulating systems of sizes and complexity that have not been before. You can run into these kind of troubles. Here it actually turned out that the fact that this system started spinning was due to some not conservative enough settings of the neighbor list, which was also shown in previous work of the Hummer lab to be able to create artifacts in large systems. And this was also causing this strange spinning behavior in this particular system. Then you can use slightly more conservative neighbor list settings, and then actually it turns out the spinning problem was solved. So here's actually another simulation now of 100 nanoseconds that we have been able to do on this system of realistic dynamics of this cell. And of course, on this time scale, not too much exciting is happening, except for still one issue that we are facing with this setup. As you see here, actually, it turns out that there's too much tension initially on the membrane, which causes some rupture. And that's another interesting aspect of building these kind of systems, that it's very difficult to, in the initial state, to put exactly the right amount of solvent inside this system. So if you put too much, which we apparently did, this will create a little bit too much tension on the membrane, and then it can rupture. Of course, eventually this gap will heal again as excess solvent can now move to the outside. But of course, this is not what we want, because this also causes some other potential artifacts of membrane proteins maybe flipping around. So we're constructing now different setups to try to mitigate this potential problem. But in principle, I think we now can state that we can simulate at least a small version of this cell for longer timescales. So of course, that is what we eventually are aiming for. I think eventually we would like to at least get dynamics on hundreds of microseconds. And then I think there's many different intriguing questions that we should be able to answer. And one of the major ones is, of course, how homogeneous are all the components being distributed? We put everything originally more or less random inside this cell, but my gut feeling is that the real organization of this cell will not be random. And hopefully our simulations will be able to tell us something to what extent they are non-randomly distributed. Maybe there is depletion or enrichment layers near the cell membrane, for instance. Or there might be biomolecular condensates that are being formed, like in many cells. Also dynamically, in this crowded environment, which molecules can find each other, on which timescales are there certain areas where things can diffuse faster than in other parts of the cell. And in general, just on which timescales and which molecules interact with which other molecules inside this very crowded and complex environment. All right, I'm asking Alessandra now, how much time do I have? Because I don't see a clock on my own computer.
It's 44.
44. Okay, then I will wrap up. So there will be time for questions. So a couple of more slides on the perspective. So we have, of course, a perspective also to couple the kind of Martini level simulations to the lattice microbe simulations at the Schulten lab, because then we could potentially, with this lattice microbe, really probe the different cell states and then probe the chemical reactions. And then once we have a new cell state, we could map it back to the Martini level of resolution, simulate it for tens, hundreds of microseconds. And from the information we get from those simulations, inspire again the assumptions underlying these kinetic reactive models. Another perspective is moving to other large complex systems. And one target that we have is simulating an entire mitochondrion, which we already did, but then only the lipids with the dry Martini version, just as a showcase that we could construct these kind of complex, highly curved membrane shapes with the TS2CG tool. But of course, now we want to also go beyond that and embed all the membrane proteins, the mitochondrial chromosome, and all the metabolites. So we're currently constructing these kind of mitoslice models. We call them models of the crista of the mitochondria with the right amount and stoichiometry of the membrane proteins. So this is also work in progress. And we also further develop our tools to be able to pack also in these highly curved geometries the right compartments, because there's different compartments inside this mitochondria with the right mixture of proteins and metabolites. So we're making good progress on that end as well. All right, I think then with that, I'm going to end by thanking all the people involved in all the work. We have a large team of people involved in Martini development, and there's many more actually that contribute to the various task forces that we have to constantly trying to improve the models and to make efficient tools to handle all sorts of systems. As I say, we work together with the Zen Schulten lab on modeling these whole cells. And then in my group, there's many people that work on the cell, but also on many other systems that I didn't have time to talk about. In particular, Jan Stevens is the main driver of this whole cell project. And then the final simulation I'm going to show is actually an animation made by Zane Thornberg from the Schulten lab, where he ported our minimal cell model into Minecraft. So you can now find this minimal cell model inside Minecraft, and you can have a look around. You can enter the cell and you can see all the components, the chromosome, the ribosomes. And you can just enjoy it, or you can even shoot it to pieces if you don't like it so much, whatever you prefer. And with that, I would very much like to thank you for your attention.
Thank you very much, Siewert-Jan. Great talk. So now we start with a question. There is Cesar. Cesar, I will unmute you so you can ask your question yourself. Please. Cesar, could you speak? Now you are allowed to unmute yourself.
Can you hear me?
Yes, please go ahead.
That's amazing, an amazing work. I mean, I couldn't imagine we can even simulate a cell at this point. But I'm wondering, what are the things that are difficult to predict from this kind of models? Because we are not simulating whole atoms on here. So would you elaborate a bit more on this, please?
Yes, certainly. And I think one answer to your question would be that we don't know, because that is, I think, the beauty if you add more complexity, because in the end, in a cell, all the processes are somehow connected to each other. And both on a small scale, so how two proteins come together, of course, depends very much on what kind of proteins they are, but it is also affected by their surroundings. And the surroundings is again affected by the larger surroundings. So it's a whole hierarchy of length and time scales that connect everything together. And to understand how that works, in the end, you need a simulation that includes all these details. And then maybe for some processes, it turns out that we could have simulated the interaction of two proteins, ignoring everything else. And then, of course, if you want to know more detail about that, it would make sense not to include the entire cell. But I'm sure, but this remains to be seen, that there will be many events that really require everything to be present, or at least it will affect the outcome. So, but what these processes are, of course, remains to be seen to some extent. But more concretely, as I alluded to in one of the last slides that I showed, I think we want to see first how homogeneous things are being distributed inside the cell. And I think for that, a coarse-grained model is very well suited, because it doesn't probably rely on very detailed hydrogen bonds that are formed, but more the overall affinity that compounds have for each other or for different kind of environments. I hope that answers your question to some extent, but it's a good question.
Thank you, Siewert-Jan. So then we have some question from an anonymous attendees that is more asking about the setting that you have done. So he was wondering, I think this question came up as before you start going more in details in the cell simulation. So for you to set the question in the correct time of your talk. In equilibrium is the first question. The second one is, are the local environment of single protein in equilibrium, or do they stay out of the equilibrium?
Oh, that's also a very good question. And I should have mentioned this more explicitly, that the cell that we are simulating at the Martini level at the moment is kind of a dead cell. There's no chemical reactivity occurring. And that is also, well, maybe at some point we can connect it to the reactive Martini scheme that we are also developing, but this will still be a long way ahead. So for the moment, we're just doing classical molecular dynamics. And so many aspects of cellular processes will not be happening in our cell. And so one way out is, of course, to connect this to the reactive models of the Schulten group, as I mentioned, because they can follow the reactions. So then at least we can have an updated composition of the cell and see how that then affects the global organization. So other than that, yeah, so simulating this cell much beyond the 10 to 100 microseconds is probably not going to be useful anymore, because on those time scales, really reactions will have taken place that will change the nature of the cell. Another part is that it indeed might be, and there's growing evidence, that you need some kind of chemical reactivity also to keep the cytoplasm not going into a glass state, for instance. These kind of things could happen. So it could turn out that our simulations show that without this chemical reactivity, eventually we end up in a glassy type of cytoplasm state. And then if that happens, of course, we can find pragmatic solutions to mimic the effect of the reaction by maybe adding some heat to certain proteins that would undergo these chemical reactions and allow the cytoplasm to become fluid again. But this is all to be seen how that happens. But those are the ways or the lines we are thinking.
Okay, thank you very much. So now we have Alexandra Farkas. Alexandra, do you want to ask your question? You can unmute yourself now. Okay, I guess Alessandra cannot speak. So I will ask the question. So she's asking about Martini 3 and the nucleic acid. And the question is if does hydrophobicity still appear when simulating interacting amine nucleic acid?
Sorry, I'm not sure I completely get this question.
So I think the question, I don't know. So she asked about Martini 3 and a nucleic acid. And the question is concerning hydrophobicity. I think I guess there was some issue with hydrophobicity before. I don't know. This is my guess. Still appear when simulating interaction between two nucleic acids. That is what I understand from the question.
Okay, well, first of all, we are currently developing the Martini 3 nucleotide model. So the simulations I was showing of the whole cell are still Martini 2 based, where we had developed a nucleotide model. So we're currently in the process of getting the Martini 3 nucleotides out. But yeah, this is ongoing. I don't recall any problem with hydrophobicity. So hydrophobicity as such is included in Martini in an effective way. So real hydrophobicity originates through entropic forces of water that gets oriented near hydrophobic surfaces. Of course, we have a lower resolution model, so part of that entropy we need to replace by enthalpic terms. So this is done in Martini. So for instance, oil-like beads and water-type beads, they have a lower Lennard-Jones interaction between them, which makes sure that they will phase separate. So that is kind of how we capture the hydrophobic effect in general in Martini. So also the nucleotides follow these same rules.
Okay, thank you. And now there is a question from Ilpo. I will try, it's a long question. I try to unmute Ilpo. Ilpo, if you want, you can speak. Oh, I guess Ilpo cannot. Okay, I will read the question. So I say, great talk. I'm now in Lapland with oh, sorry, it doesn't have a lot of band. Sorry, I just asked now. So in this first question, in the simulation business, we are aware of the I don't know how to pronounce these things. YPWOGO principle. Do you know it? What is Ilpo speaking about, Siewert-Jan?
No, maybe if I can see it in the chat, but I don't have, I'm still sharing my screen. No, I'm not exactly sure what he.
I can just.
Maybe it's something like what you put in, what you get out.
Yes, I guess so, but
oh yeah, it says there even.
Yeah, it's what you put in and what you get out principle. Yeah. Sorry, I was not able to read it from the question. That so you will say, okay, in the simulation business, we are aware of the what you get in and what you get out principle that we try to avoid. That is how to sample the system extensively such that the result would not reflect the initial states. However, in this huge system, the times needed for sampling will also be huge, yet the simulation time remain quite modest. Your plan to overcome this issue?
Yeah, that's of course a very valid point. Thanks, Ilpo, and enjoy Lapland. Yes, and certainly even if we can run 100 microsecond of this whole cell, the final state of that cell will still be very much biased by the initial state. Now, there's a couple of answers to that. First, complete equilibration is not maybe a real target because, yeah, as also discussed, you need chemical reactions to keep a cell actually out of equilibrium. So we probably don't want to reach real equilibrium in the traditional sense of MD simulation. Second, I think firstly we will be looking at how do you say that, trends of moving away from our initial state. This will already be informative. We start with a completely random state and moving away from that already provides this possible clues as what will happen inside a real cell as well. The third thing I want to say is that we consider this kind of setup, but also the simulations itself as kind of a living simulation, meaning that we will constantly try to improve and make the simulations more realistic, either by having improved versions of our force field models, but also integrating more and more experimental data. And hopefully this can be iteratively, we providing clues to experimentalists to look for certain things, and then experimentalists providing us with information that we can better build our initial models, because there's more information about certain proteins that are likely to interact together, so we can already put them close together in our initial state. So that's one way that I think over the years we will aim to get more and more realistic overall models of this minimal cell or whatever other cell we eventually want to simulate.
Yeah, I think in your answer, thank you, Siewert-Jan. I think in your answer you also addressed the second question of Ilpo, but please, I read the question and confirm it. Also related to sampling issues, your view as to how to pick biologically relevant question in this setting.
Well,
I guess you partially address it.
Yeah, I hope I did to some extent. I think understanding how a cell is organized is what many, even also experimental biologists working in this field, ultimately would like to do. And they do it with high resolution supra microscopy or with other fancy techniques. And we do it from the computational side, but eventually integrated, this should give us more understanding of how cells function at a molecular level, and can also be instrumental in building synthetic cells from the bottom up. So I think, yeah, that is also my major driving force, I would say.
Okay, I think we have a lot of other questions, but I will suggest that the people can follow up in the forum for the following up question. And because it's over the hour, so I want to close this section. And if you stop sharing, please, Siewert-Jan, I can just announce the following up. Thank you very much. I can just show. So I want just to announce the following webinar. So we will have the 8th of April. We will have Marco Giulini that will be speaking about modeling antibody in the post-alpha fold area, where we are now. And then I want also to announce that Bioexcel is now will run in May, exactly the 21st to 22nd May, an ambassador-driven workshop in Sofia in Bulgaria, where we will have training concerning GROMACS, performance and advanced sampling and introduction to GROMACS, PMX and ADOC. And the deadline for register to the events is the 15th of April. And finally, I would like to thanks a lot, Siewert-Jan, for joining us for this webinar. Thank you very much. And all the attendees for being with us and for all the questions. And now I close the section. Thank you again and see you next time. Bye-bye.
Thank you.